the bio and the... Tonight's speaker, Dr. Jovana Jasso-Martínez, is a Peter Buck postdoctoral working at the Smithsonian as a researcher at the Museum of Natural History. Dr. Martinez received her Ph.D. from the Universidad Nacional Autónoma in Mexico, May of 2022. Her research includes systematics and phylogenomics and evolution of insect groups, mainly Hymenoptera. The project that Dr. Martinez is conducting at the Smithsonian focuses on assessing parasitoidism strategies as promoters of shift in diversification rates in the parasitoid wasp family Braconidae, using ultra-conserved element data in a phylogenomics framework. So in essence, specimens preserved in biological collections have become one of the most important sources for obtaining genomic-scale data to perform phylogenetic studies. This is particularly useful when using extinct or rare species, as well as taxa that are otherwise difficult to collect. Implementing NGS approaches for insect museum specimens is challenging, mainly because many of them are considerably small and the amount of DNA recovered is considerably low. In this talk, Dr. Martinez will show the potential of using museum specimens to perform phylogenomic approaches, employing as a model of study the parasitoid wasp of the Braconidae family, and using a substantial amount of genomic information from considerably old and pinned museum specimens. So, having said all that, I will now turn it over, and Dr. Martinez, thank you for being with us tonight, and we appreciate your taking the time to present for us. So we'll let you go ahead and do your talk, and if people have questions, sometimes people might ask during the presentation, but mostly at the end, we'll see about that. But at any rate, thank you, and I leave it to you now.
Hello, everybody. Thank you so much for the invitation to give this talk. So yeah, I'm gonna talk about past and ongoing research that I am performing at the National Museum of Natural History, or NMNH. So I hope you enjoy this talk. Most of them housed in museums. Natural history collections are an invaluable record of the diversity and evolution of life. In biological collections, there are deposited billion specimens which can be considered as a long-term source of data for the comparison and discovery of taxa, as well as to provide a basis to perform different studies, including macroevolutionary, population, environmental, and morphology-based. These biological collections are a global network of research for educators and students in different areas of knowledge. And very important currently, the digitization of material that is deposited in collections greatly helps to keep this network working. Most of the specimens deposited in collections include associated data, such as geographical collection events information, such as collector, year of collection, and even host records in the case of parasitic groups. Also, collections are a very important repository for fossils, extinct taxa, as well as extant taxa, both rare or difficult to collect. Such as species with very restricted to a geographical area or endemic, or taxa for which is not possible to make directed field work given their unknown biology or even their diminutive size. Also, the material deposit in collections is a very important source to perform morphology-based studies, such as the description of new taxa, with several new species discovered in collections and then described just recently. And more in the mood of this talk, specimens in collections are currently one of the main sources of tissues for DNA extraction and its subsequent processing. Next generation sequencing techniques have revolutionized the acquisition of genetic data due to the advent of the production and assembly of millions of short DNA fragments, which is particularly beneficial when working with museum or historical specimens that in many cases are old and sometimes are not in the best state of preservation, which conduct to the degradation of DNA, as is the case of many groups of insects. One more complication in the case of some groups of hymenopterans is their very small size, which conduct to the obtention of only a few amounts of DNA. All these have opened the field of museomics, which is the study of genomic data obtained from historical and DNA from specimens in museum collections. In the last few years, museomics-based research has increased significantly. Just last year were published near 30 museomics-based works to answer questions in the population genetics, environmental sciences, and systematics. But, sorry? Sorry, I think.
It's okay.
Okay. Continuing with this, what kind of genomic information using NGS techniques it is possible to obtain from museum specimens? In particular, target enrichment methods such as anchor hybrid enrichment and target capture of ultra-conserved elements or UCEs have demonstrated a higher success rate over enzyme-associated when using degrade or low-quality samples as old and dry museum specimens. All methods, to be honest, all methods have their advantages, and the use of one or other needs to be evaluated depending on the question we want to answer. Just an interesting fact, particularly when working with UCs, it is also possible to recover mitogenomic information as secondary product, in many cases allowing recovering complete or nearly complete mitochondrial genomes. So having this in mind, the potential of UCs for phylogenomic reconstruction, and given that the available specimens we had to make studies about Braconidae were museum specimens, in many cases very old ones, we decide using this technique to reconstruct, UCs to reconstruct the phylogenetic relationships of the members of the family Braconidae. Parasitoid wasps are free-living insects as adults. Females lay their eggs in the bodies of other arthropods, mainly insects, so the larvae develop consuming the host and further causing its death. Parasitoid wasps are an extremely diverse group and economically important, since it contains many species that are used as a biological control agents of plagues. Specifically, I work with the parasitoid wasps of the family Braconidae. This is the second largest family in Hymenoptera, comprising over 21,000 described species, although it is estimated that this species richness could be the double or even more. Braconidae are beautiful wasps. They can be very small, although there are very big species. They live virtually in every ecosystem. They are all over the world, and they are very abundant. These wasps can be endo or ectoparasitoids, which mean that the eggs are laid into or on the host, respectively. Also, they can be coinio or idiobiont, which means if the host is allowed to further development or not, respectively. But even though Braconidae is a widely known group given its ecological and economic importance, the superspecific classification of this family has constantly changed, in part due to the high levels of morphological convergence found among its members, and also because the lack of comprehensive taxonomic and molecular sampling in previous studies, having as a main goal reconstruct higher level phylogenesis of Braconidae, leading to obtain weakly support phylogenetic hypothesis. Given this, the establishment of a more stable classification for Braconidae is one priority that can be reached by the use of integrative tools that allow making better informed taxonomic decisions and also answering different evolutionary questions related to their extraordinary biological and morphological diversity. So making use of museum samples, many of which were significantly old, we reconstructed a phylogeny of Braconidae, including most of its recognized subfamilies using UCE data. This was the phylogeny of Braconidae with the most comprehensive taxonomic sampling for the family, including taxa for which their placement has been problematic in previous studies, such as the members of the the previous recognized as subfamily Trachypetidae, and Apostis spengai, which have been at some point elevated to family level or placed into the sister family of Braconidae, the Ichneumonidae. We recover a monophyletic and well-supported Braconidae, including both of these taxa, and we also recover with the strong support phylogenetic relationships that were recovered in previous studies using other sources of molecular data or morphology exclusively. Since we use museum old samples, I was interested in investigate if there was a correlation between specimen age and the amount of genomic information recovered, specifically the number of UCE loci. A correlation between the specimen's age versus the number of UCE loci was recovered, which means that for older specimens, we recover significantly less UCE loci in contrast with more recent samples. So I analyzed separately those samples whose libraries were enriched with different dates, versions, which is important since both versions date versions capture different amount of UCE loci, and in both data sets we obtained similar results. And for the oldest sample in both data sets, 82 and 90 years old, we've recovered 130 and 818 UCE loci respectively, which in both cases is a considerably good amount of information to be used in a phylogeny. As I mentioned, robust phylogenies help to answer with a better confidence other evolutionary questions. For Braconidae, it is particularly interesting to investigate the evolution of its different parasitoidism strategies. In this respect, it has been proposed that within Ichneumonidae, superfamily Braconidae belongs to, there has been a transition from ectoparasitoidism, idiobiosis of concealed host, to endoparasitoidism, coinobiosis of both concealed and exposed hosts, mainly based on the consideration that eggs of endoparasitoids of exposed hosts are protected from the external environment. And at the same time, there is a better exploitation of resources for the parasitoid offsprings since the host is not paralyzed in the case of coinobions. This hypothesis, in fact, was never assessed in a phylogenetic context, but until 2021. In this work, authors tested the hypothesis of these transitions for Ichneumonidae, including Braconidae and Ichneumonidae, and based on ancestral states reconstruction, they recover as the ancestral state of the whole superfamily, the strategy Idiobion ectoparasitoid, with multiple subsequent transitions. However, for Braconidae, the results were differently recovered depending on the sampling used. Considering this and using the comprehensive phylogeny of Braconidae we recovered using UCE data, we reconstructed ancestral states again for both endoparasitoidism as well as for coinoidiobiosis. And we recovered coinobiosis endoparasitoidism as ancestral, at the ancestral state with multiple subsequent transitions. These results show that coinobiosis endoparasitoidism and on the other hand, idiobiosis ectoparasitoidism are strongly correlated, although there are few cases in where this correlation is kind of broken, such in the subfamily Rhizopolinae, whose species are coinobiont ectoparasitoid. Although in general now we have a better understanding of the Braconidae phylogeny as well as of the evolution of its parasitoidism strategies, other questions still remain. A striking feature within Braconidae is the pronounced difference in known species among its lineages. For example, subfamilies with some having fewer than 50 species, such as Jasso-Martínez and Rhizopolinae, other a few hundred, such as Agathidiinae and Rugadiinae, and yet others with more than 2,000 species, such as Dorictinae and Microgastrainae. This marked difference in species richness among Braconidae lineages could be explained by the age of different lineages, although it could be also explained by diversification bonds promoted by shift in parasitoidism strategies and linked features, which are issues that have not been explored for Braconidae in a well-supported phylogenetic context. So as preliminary results of my research at the NMNH, I am performing I am performing phylogenetic reconstruction, and I have recovered a more comprehensive and well-supported phylogeny of Braconidae, which include more than 600 Braconic taxa, as well as 4 out of the 41 recognized families for Braconidae. All new data was generated using specimens that were deposited in the collection at the NMNH. One of them was a tiny and more than 100 years old specimen for which we were able to recover a considerable amount of genomic information. At this point, we have also generated a very preliminary dated phylogeny of Braconidae using 6 fossil calibrations, from which 4 belong to different extant Braconidae families and the remaining 2 to extinct taxa. We estimated the origin of crown Inomonoidea, superfamily Braconidae belongs to, in the Middle Triassic, other than the most recently estimated origin for the group. Specifically for Braconidae, we estimated the age of its most recent common ancestor in the late Triassic with an age of 2,022 million years. And using the preliminary dated phylogeny of Braconidae, I explored the occurrence of differential diversification among clades within Braconidae. For 4 of the Braconid clades, we recovered a faster rates of diversification. These clades include 2 Coinobion families, whereas the other 2 clades represent Idiobion families. Based on these very preliminary results, it seems that faster rates occurs in clades that display different strategies, and that also that these families attack very different hosts. So at this point, we haven't found a correlation between faster diversification rates with respect to biology of these groups. However, these results are very preliminary, so more exploration about these issues is still need to address these other questions. And finally, I would like to highlight that museum collections house millions of specimens with potential to be used in different studies. In particular, for phylogenomic studies, material in collections allow the opportunity to increase taxon representation, including rare or difficult to collect taxa. Also, independently that the amount of genomic data is correlated to the age of specimens, the information obtained is highly informative, and particularly target enrichment approaches are suitable to perform museomics-based research. And finally, the genomic information obtained from museum specimens is usable and informative to perform macroevolutionary questions in megadiverse taxa such as parasitoid wasps. And finally, I would like to thank all these institutions that have helping me to perform these studies, and also co-authors and other colleagues that also have helped a lot to develop this research on Braconid wasps. Thank you so much. So I will be happy to take your questions.
I have a question for you.
Yeah.
It was a very interesting presentation. Thank you so much.
Thank you.
At the beginning, you were talking about combining DNA. I'm not sure I understood quite well. If you didn't have enough collectible material from one set, you could combine it with another in order to get the data that you needed. Did I get that right?
No, I mean, when the NGS approaches started to be used for phylogenomic studies, it was necessary to use a very fresh material, for example, in ethanol or material recently collected. But with the advantage of development of NGS techniques, it was possible to use a material that was already deposited in collections. Most of this material in many collections is pretty old. So I was able to go to the collections and find these the specimens that we were lacking in previous studies, and recover a high amount of genomic information to be used to perform phylogenetic studies. So I don't know if I answered your question.
So basically, you took this genetic material from one species rather than a collection of that species and another species to get enough DNA.
Yeah, but I mean, I didn't mix, the DNA from different species. So I just used this DNA to extract genomic information and then used them, in a phylogenetic reconstruction.
It's got to be so difficult to get old material for 100-year-old species. I mean, gosh, it's got to be dried up.
Yeah, I mean, it has been, in fact, at the beginning, it wasn't easy because, lab protocols are standard protocols. So we needed to make some changes in some of these protocols. But it is these NGS techniques, they are so useful, and yeah, in fact, it is possible to do this, and this is impressive that from a tiny specimen, one can obtain this huge amount of genomic information. So yeah, that's huge, and also recover this huge amount of genomic information for very old specimens. It is just like, right, it's impressive, right?
Yeah. Thank you so much. That's just unbelievable.
Yeah. When I started doing this, working with this group of wasps, many of which are super small, I was wondering, is this gonna work? And it did it. So yeah, of course, we have a lot of other previous studies to compare, because of course, you can get genomic information, but the data that you obtain makes sense, like in a phylogenetic context. So that's why it's very important to have previous phylogenetic hypothesis. And yeah, our phylogenetic hypothesis makes biological sense, and so that's how we can test if this genomic information is already usable, right? But yeah, it's a lot of work, but it is worth it.
For us non-DNA types, can you briefly go over the steps that's required to do these things?
Sure. Also, you have your specimen, right? You can use a recently collected material, or as I mentioned in this talk, a material that is already in the collections. So then you need to extract DNA. So it depends how you are going to extract this DNA. It's going to depend on how big is the specimen. For example, if you are working with a super big specimen, like 1 centimeter or something in the case of Braconidae, you can take a single leg. Or if the specimen is super small, you can take the whole specimen and then use protocols of extracted DNA, for example, like enzymes that are gonna break the cells and extract the DNA. And once you have your DNA, you need to construct a library, a genomic library, which is basically fragment this DNA in very short pieces, and then in the lab put some other sequences that are gonna help to sequence this material. I don't know how to explain that in it's a protocol in where you need to put adapters to sequencing platforms. So the sequencing is gonna be helped by these sequences or adapters that are attached to the DNA fragments of your specimens. And then for specifically for UCs or any other target capture protocol, you need to, I mean, there are some baits, or which are a few short fragments also of DNA, but that are recovered from aligning genomes from, in my case of Hymenoptera. And these little pieces of DNA targets ultra-conserved elements or ultra-conserved DNA sequences in the DNA. And then we need to perform other lab protocols for extract these ultra-conserved sequences. And then all this information is sent to a company that sequence everything. And once we get our data back, we need to perform different bioinformatic pipelines to try to recover bioinformatically these regions, and then perform DNA alignments, and then reconstruct phylogeny. So basically, that's the pipeline from the specimen that is in the collection to get phylogenetic reconstruction. So I don't know if I answered your question.
Very nicely. It sounds so easy, right?
It sounds so easy. I mean, when I started doing this, since it sounds so easy, I started doing that. But then when I was actually doing it, it is not that easy. But I mean, as I mentioned, it's worth it also to try to answer these very interesting evolutionary questions. So it is impressive that one can try to give these inferences from very tiny specimens and from this huge amount of DNA. I mean, that you can't actually see, right? Like, I mean, it's different that you can see your specimen in the mic, in the scope, but you can't actually see your DNA. So it is still kind of, yeah. Right?
Very nice. Thank you so much.
Thank you.
So I was going to ask a similar question. Dr. Hansen beat me to it, and I appreciate your reply to that. So in your case, you're taking a leg, let's say, off of an old specimen, and is that what you send that leg the way it is over to the lab, or are you going to be doing some of the molecular work yourself at the beginning of it?
I do all the molecular work by myself.
Okay. Right.
The only thing I can do is sequencing, like actual sequencing. So,
okay.
but all these lab protocols, from extracting DNA, library preparation, and enrichment protocols, I do that by myself.
Okay, that's interesting. How many specimens are you working with at a time? I mean, is it you using a lot? Imagine.
I think, I mean, only for this project with Braconidae, I have processed like 4,000 or 5,000.
Oh, wow.
Do you
That's
send that
amazing.
many eggs in? Of legs in?
Yeah, I mean, no, I said 1,000, no, 100. Sorry, that's a translation problem. But yeah, no, I mean, it is still a lot. I mean, working with this kind of protocols it's a lot of work. But yeah, I mean, like 500. Sorry, but it is still a lot, I think.
Great.
We have an interesting question in the chat box. So there's a lady who's online. I believe she's still on. Yes, and the young
That's a
child, Gwen. Rachel, do you want to ask the question, or shall I?
So probably you would like to try to give me your an ID for this specimen. Not sure.
So
okay, I can.
let me.
The research is very amazing.
There was a good question from the kid here. Gwen. Gwen, how old are you, honey? Can't hear you.
How old are you?
5.
5. Wow. So
Mhm.
when Dr. Martinez mentioned that using the ultra conserved DNA elements, they were able to know that some of the ancestors of these wasps lived during the Jurassic and Cretaceous periods. Now, Gwen would like to know how the Braconidae and Ichneumonidae wasp could have survived the extinction events that killed the dinosaurs.
Wow, that's a super interesting question. I have never had this kind of questions in my talk. Yeah, I mean, I love how curious are kids. They make the most interesting questions always,
Yeah.
I would say. To be honest, I think it was for the use of niches. I mean, the massive extinctions that killed the dinosaurs. Dinosaurs were huge animals, right? So I basically think it was survived because they were tiny and they were not only, for example, in the places in where dinosaurs used to live, these wasps are all over the world. So I mean, they were not restricted to an area. So I think that's why, and also they used different, super different resources to survive. So I think that that combination helped them to survive to these massive extinctions. And also they are super diverse. So I mean, I think in general for mega diverse groups is quite easier to survive to massive extinctions in comparison with other groups that have only a few species and that are, for example, endemic or something like that. So yeah, that's what I think that helped them to survive.
Thank you so much.
You're welcome.
Thanks.
You're welcome, and thank you for attending my talk.
Thank you, little Gwen. So there's a picture in the chat, I think, that was posted by Rachel, and
Monoid wasps.
that's Gwen's mother, right? And there's a small wasp in the picture. If you click on the chat, Jovana, do you see it?
Yes. Yeah, I mean, I can see the wings, so that will be super important to try to give an ID. But in fact, I am not sure if it is an ichneumoniid. Probably it is a braconid. It looks like probably a Brocadaini for me. But I don't know if this was, I mean, it was at night because I mean, Brogadines are nocturnal. So yeah, I mean, but it is definitely an ichneumoniid.
Oh, great. Thank you.
Was it at night? Was it at night, Gwen?
I think, yeah. It was on our ghost blow up walking around. Like,
yeah, on a Halloween decoration, right?
yeah.
Okay.
Oh wow.
So
Yeah.
it came to the light, basically, right? Yeah. That would make sense. Yeah.
Yeah, but also that now I'm having a look at the antennae. Probably it is an ichneumoniid, but yeah, I can't confirm because I can't see the wing venation. But I mean, it could be an ichneumoniid or a braconid, of course, but probably Rogadini subfamily. It looks to me like that. It is beautiful.
Okay. Well, thank you.
Thank you.
Another question that came to my mind was that has anybody tried to extract any DNA out of amber? Because I know that braconids have been found in amber, and frequently at that. All kinds of little wasps have been found in amber. Is any of that material actually salvageable to get any kind of sequences out of?
I don't know anyone that is trying to do that. I mean, there are plenty of protocols with other groups like, I mean, mammals mammals mostly, that try to recover information from this historical DNA. But I don't think anybody's trying to do this with fossils in amber.
Yeah, I've seen some very well preserved insects in amber and even larvae preserved in amber. But I don't know if there's anything that's salvageable out of that. It doesn't seem like it always is, but
you need 500 wouldn't you?
500 what?
Examples of samples of insects in amber, according to the speech to the seminar tonight. You need quite a few, right?
Oh, you would need quite a few, yeah, right. Imagine so. But Jovana can tell us about that. Is that the case, Jovana?
The case of for extracting DNA from fossils?
Yeah.
I mean, as I mentioned, I don't know anybody that has been trying to extract DNA from insect fossils. I don't have any other background.
Would you need a lot of specimens? I think you mentioned for museum specimens you can use one leg.
Yeah, it is possible to use one leg, but
Okay.
I mean, yeah. So it's not that we need like many specimens that represent one species to get DNA. It's only a single specimen per species.
Right. Okay. So are you going to continue working with this group, or do you have another group that you work on besides Braconidae?
I started working during my bachelor's and my master. I worked with LEPs, population genetics with butterflies, and then I changed to braconids. I mean, also it was super interesting because there is a bridge, right? Because braconids attack many groups of Lepidopterans. So that was interesting for me. And also I have a bit of experience with orthopterans. I have some works with orthopterans, but I mean, braconids is a very interesting group. There are many evolutionary questions that need still to be addressed. So I would like to keep working with braconids, but also, I mean, something that I like about this kind of research is that these evolutionary questions can be addressed in different groups. So if I have the opportunity to work with a different insect group, I will do it. But I mean, braconids, I like them. I have been working for a while with them.
Your video early in the presentation with the wasp attacking the larvae was very nice video. And I wondered if you could reiterate the definitions that you gave us earlier about kino and idiosymbiont. Could you just mention again? I was trying to remember them in my head.
Braconids and other parasitoid wasps can be endo or ectoparasitoids, which means that the eggs are laid into or on the host, right? So endoparasitoid wasps are generally also coinovions, which means that the host is allowed to further development. I mean, once the host receive the egg, I mean, the egg is laid into the host can still be like living a normal life. They can feed and they can move, and sometimes the caterpillar can pupate, but at the end it's gonna be killed because the presence of the parasitoid wasp, right? And in contrast, the ectoparasitoids, which lay the egg on the host, they regularly are idiovions. Idiobiosis means that the host is permanently paralyzed. So once the egg is laid on the host, for example, a caterpillar, stop moving and stop development. Is completely paralyzed, and at the end, it is killed by this interaction.
Thank you very much. It makes sense too, because the word kino in Greek means moving, right? So I was trying to piece it together, but I appreciate that. Thank you.
Yeah, you're welcome.
Does that suggest if the caterpillar is not able to eat anymore, that it won't provide food for very many parasites?
Yeah.
Whereas if it was allowed to continue to eat, it could support a lot more parasites. Have you been finding that's the case?
Yeah, in fact, it is a super interesting question because, I mean, both the strategies have other associated features. And idiobions, which, I mean, when the host is paralyzed permanently, as you correctly said, it is not allowed to continue feeding, right? So idiobions have a faster development in contrast with coinobions, because, I mean, coinobions can exploit the resources that the caterpillar is getting because it's allowed to further development. So yeah, I mean, there are trade-offs. Yeah, I mean, since the caterpillar, I mean, the host that is permanently paralyzed is not allowed to continue feeding, this has an effect on the parasitoid that is attacking it. So I mean, the eggs are smaller, they develop faster, they form the pupa faster. So yeah, these are kind of trade-offs that are found between coino and idio strategy.
Very interesting.
Mm-hmm.
Some more questions for our speaker. Anybody? Somebody might. Ed, you usually have a couple questions. Anything on your mind?
Mute. I'm unmuted. No, I
There
don't
you
have
go.
this interesting presentation. All these insects that actually have survived, you know, over many, many eons of time. And whereas, as you mentioned, the big extinction that occurred, how many million years ago? Was it 65 million years ago, something like that?
Yeah.
The big, yeah. Yeah, then these little insects that just by their numbers survive, right? And they're so small, they sort of do, they manage to survive. You know, it's sort of, that's sort of interesting to me, I guess. Yeah. Well, it's interesting research.
Thank you.
I don't have any other comments to make, really, but I've got I was interested in Braconidae and also in Ichneumonides. Would you think the Ichneumonides would be also interesting to study in the same context, or what?
Yeah, I mean, in fact, Ichneumonides are being studied in the same context by mainly, I mean, for a lot of colleagues of mine, but also mainly for by Bernardo Santos. He was working at the NMNH too, as I am, and now he's working in Germany. He is a researcher in there. So he and I mean, we kind of make similar research, but on different groups. And also, I mean, Dr. Barbara Sharanovsky, I don't know if you know her, she has been working also with these kind of questions for the whole Ichneumonidae, not only for Braconids or Ichneumonids, but for both of them. So yeah, I mean, yeah, Ichneumonids are being investigated too in this same context.
Oh,
Mm-hmm.
interesting. Okay, that's all I have to say at the moment.
Thank you, Ed. All right. So Anybody else would like to comment or ask a question? So, Jovana, you are going to be our honorary member for 1 year.
Oh, thank you.
All right. So it's our gift, small gift to you, a membership for 1 year for being speaker tonight. And you also receive our journal that we put out. So you get to read our Maryland Entomologist, it's called, and newsletters you will receive. In fact, I should have sent you, but I don't think I did send you the. Did I send you the Phaeton, the newsletter? I gave you the announcement, right? Okay,
Yeah.
I'll forward you the newsletter also, but it came out after the announcement did, so I'll forward that to you. But if you could email me a mailing address that I can have the journal mailed to you,
Sure.
that would be great. Just text me or email me that information, okay?
Thank you so much. Thank you.
Thank you. Thank you. It's the least we can do for you, and we greatly appreciate you taking the time to speak to us tonight. And it's a very interesting subject. And one thing I have to tell when I speak to my students, and I always mention that anymore, the research in biology is oriented in the molecular level. Everything is molecular nowadays because that's where a lot of these questions can be analyzed and answered and figured out. So when we were, myself and some of the members were in school, it was whole organism biology, right? But now we're dealing with strictly molecular in most cases. So and it's interesting because, of course, with molecular biology, you can go in many directions and answer a lot of questions that way too. So anyway, what more can we say? Good luck with your research. I hope it continues very well for you. And when you receive future announcements, I hope you join us again and attend one of our meetings when you can. And I look forward to meeting you sometime if you go to the ESW. Do you attend ESW meetings at the museum?
EW meetings. I don't know what kind of. I mean,
Washington, Entomological Society of Washington. At the
yeah. I was a speaker in that one. I think it was last year. I don't even remember, but yeah, every we have a meeting every first Thursday.
Yeah, but I was saying to you before, Thursday is a bad day for me. Otherwise, I would go more often. But anyway, maybe I'll see you there in January.
Okay, I hope so. Yeah, I mean, I hope so because I mean, for Christmas I go to Mexico, and sometimes in January, like at the beginning of January, I am not here. But
oh yeah, right.
But
well, I can't blame you. It's nicer in Mexico in January. All right. Well, we're gonna.
Can I ask one more question?
Oh, sure.
Fred?
Please.
Actually, two. Really enjoyed your talk, and I was particularly interested in that last photo. Is that the insect collection of the Smithsonian?
Yeah, the one. Yeah, it is the Lepidoptera collection of the Smithsonian, the one I show like in one of the last slides.
And so
that was my collection, Dr. Hansen.
Up in your bedroom.
In my bug room.
I can share again probably my screen so everyone can see again this one, right? Yeah.
And does it go all the way back to the far wall with, I can see shelves being and how many million pinned insects do you think they have?
I mean, for Lepidoptera, I have no idea, to be honest. I have no idea. But for Braconidae, there are like 2 billion specimens. including pins and ethanol specimens.
How many?
2.5 billion.
Billion?
Yeah, it is huge.
Billion?
Yeah, including ethanol.
The ethanol, right. Holy Moses. Yeah.
Yeah, it is huge. Yeah, there is a lot of ethanol material that is unsorted because I mean you need an army to do that. Many people.
Literally.
Well, are you the army?
Yeah.
Yeah, I mean, I in fact now for an alternative project, I am sorting some specimens, and yeah, I mean that's a work that can take a whole life. Of course, I mean you know about that. It's just yeah. I have been working on that recently.
If I might add something, Dr. Hansen, what you see here is one corridor. Now, this is, if you can imagine, there are at least, 20 corridors like that.
Going all the way back as far as
all the way back. Yeah, and these are movable stacks. It's just like at the new way they do libraries and they have movable stacks. They can move the entire row that you're looking at, a compacting system. So they've pulled out these drawers so they could take the picture and lay them on the floor. But normally when everything's closed up, they can actually move the whole row and then access the next row behind it, you see? So they save space with a compactor system. It's an amazing collection, and it's so big, of course, what you see, and I've described like 20 rows of movable stacks. I think it's like that, right, Jovana?
Yeah, they are movable stacks.
It's so interesting.
But then they have also an equally large amount, almost, maybe not quite that many, but at the Suitland Annex, which is south of Washington. In the Suitland Annex, they have the mosquito collection, and they have all the Saturnid moths and other things too. So they have a big portion of the insect collection in Suitland at that building, which is a research building and not open to the public, except for special occasions, special tours, right? And not to mention, they have a lot of other things at the Suitland Annex besides insects. I mean, for example, Teddy Roosevelt's mammals that he collected in Africa on his hunting expedition. He shot 1,200 animals, the brave hunter that he was. And those were brought to the Smithsonian, and they're all at the Suitland Annex.
1,200.
How many insects did he shoot?
Millions.
Okay. Any more on that?
Actually, yes. There were insect specimens that were shot. That's actually how the Ornithoptera came to be called bird-wing butterflies. The first specimens of them were literally shot out of the sky. So, they have specimens in the British Museum that are full of tiny pinholes because they used to use very, very fine-grained shot. They used to use, I think they used to call it dust shot from very, very small bore shotguns. And that's how they collected specimens of birds originally, because many of them, if they were subjected to a full shotgun blast, all you would have is a cloud of feathers. So they developed these very small bore shotguns to actually collect birds with. And some of these butterflies were literally mistaken for birds at a distance and shot out of the sky.
Well, I think what they did not have nets that were long enough and big enough because they fly in very high places and inaccessible off of cliffs and stuff like that. So it became possible when they first saw them. They were so taken by the giant butterfly that they brought them down with, the first specimens with the shotguns, as you mentioned. So, but I think then later on, of course, they got big nets and other ways of catching them. But The first
think,
specimens were taken that way.
yeah, I think Miss Rachel had another question that came up regarding one of the specimens that was shown in the presentation. Regarding the, I guess, the tail, the long tail on the bee.
Yeah.
That's an ovipositor.
Yeah, it says, thank you.
And
when was wondering what is the pursuit of the longest stinger of the Braconidae? Yeah, I mean, it is a long ovipositor. The wasp that is at the beginning of presentation is a Doric tiny wasp, and they attack wood-boring beetles. So they need this super long stinger to go like into the
wood.
Yeah, into the wood, and then deposit the eggs in the beetle that is inside the trunk.
Wow.
Yeah.
We had no idea.
We have some specimens of these ichneumon wasps in this country where the lengths of the ovipositors, the long tails, are as much as 5 and 6 inches. And they can detect their prey species through that much solid wood and literally bore that tail into the wood. And they'll lay the egg either directly on the grub of the insect they're parasitizing or in the tunnel that the insect grub is crawling around in. And when the wasp egg hatches, it does a number on the grub that's in the right in the solid wood.
Yeah.
Wow.
Thank you. That's amazing.
Okay.
One other question. And that was about the extinction, the fifth extinction. I've been reading Elizabeth Colbert's work on the sixth extinction. I highly recommend it for everybody. She's a reporter, or she's a writer for the New Yorker, and very scientific, very interesting, and has a number of things that she brought out that led to the to what got extinguished in the fifth extinction. That's when the meteor hit, and you mentioned basically this as well, and you suggested that the reason that we still have those wasps is because they were widely spread. And so the meteor only took out some of those more or less in the New World. Have you found any other things, particularly across species lines or across family lines in the wasp group?
I mean, if I have find other reasons for they to survive this extinction.
Yes, she suggested that in many cases it's where the eggs are laid. If they were laid deep in the ocean, they might have survived. If they were laid in the surface or in twigs and etc., they were extinguished. So I'm wondering if there's other things that you've picked up from your work, like maybe some ichneumons survived differentially than the Braconidae or whatever.
I don't know if I have an answer for that, because I mean, at least the pattern of the species richness of both families is quite similar. I mean, Ichneumonids is the most rich, the richest family within Hymenoptera, and the second one is Braconidae. So in fact, I mean, they are quite like, they have like a similar richness, and they have, but I mean, one difference, I am not saying that this helped them to survive, not, but I mean, something that is interesting is that I told you that a Braconidae, there are these different strategies, like as idiobions, coinobions, phytophagous species, etc. But ichneumonids, they are nearly all of them coinobions, so they allow further development. So I mean that's a difference, but they are. I mean, I'm at least now like the pattern we see is that they have like a similar species richness and probably similar diversification patterns. So yeah, not sure if I.
Yes. So you haven't detected any differences in richness, species richness. I was wondering if you had, or if you even had the chance to look at it. Maybe.
No, between braconids and ichneumonids, not really. I have concentrated my research on braconids. But yeah, I mean, that would be interesting that try to answer these questions, not on the separate families, right? So, but they as a whole, like a super family level, that would be pretty interesting, I think.
Yeah, would be.
Yeah.
We have to have you come back in a year or two and see if you've come up with anything else like that.
Yeah, everything together. That would be amazing, in fact.
Wouldn't it though? Yeah.
Wonderful presentation.
Yeah, thank you so much.
Thank you.
Thank you.
Thank you.
The kid says thank you. That's good.
Very nice.
Okay.
Rachel, I sent you a message. Did you see it? On the chat. So take a look. All right. Well, Jovana, thank you again.
Thank you for the invite.
You're welcome.
I had a great time with you.
Good. That's important. I'm glad to hear that. We asked you a lot of questions and kept you on for a long time. So, and we're gonna have a short business meeting. So, anybody who wants to stay is welcome to, of course. Otherwise, Jovana, if you can, when you get a chance to send me that information, and you don't have to stay, but we thank you again. And if you did want to stay, that's okay, of course, too.
Okay. Thank you so much. So
okay.
I will send you that information tomorrow, probably.
No problem. Okay.
Okay.
So,
thank you so much, everybody.
Muchísimas gracias.
De nada. Fue un placer.
Igualmente.
Gracias.
Thank you.
Thank you.
All right.
Bye.
Thank you. Bye-bye.
Que le vaya